Okay, great. Hello, everyone. Welcome to Biography. My name is Burhan Gharaibeh. I am a teaching professor at the University of Pittsburgh. And for this module in biography, we are interviewing Dr. Jonathan Wisco from Boston University Chobanian and Avedisian School of Medicine. John's specialties include neuroanatomical research, and he is a mentor on lecturing, lab organization, and administration. John is the author of the paper entitled The Effect of Age, Genotype, and Diet on Hippocampal Subfield Iron Dysregulation and Alzheimer's Disease Biomarkers in an ApoE mouse model. The paper was published in the European Journal of Neuroscience in June of 2022. Welcome, John. Thank you for joining us.
Thank you for having me. I appreciate being here. Would you like me to address you as Burhan?
Yes, please. Yeah. And
Great.
can I
Okay.
call you John?
Oh, please, 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 please. Jonathan
Oh,
is too many syllables and Dr. Wisco is too formal.
yes, thank you. Uh, John, I was wondering if you could tell us, as a non-specialized audience in neuroanatomy and neurophysiology, about the rationale for conducting this study and the main results that you and your colleagues have found from the data.
Sure. Let me go a little bit into the background of why the study is important. So, in Alzheimer's disease, which is the most common of the dementias that we know about clinically, there is an iron regulation issue that is happening in the neurons. And the neurons are the cells in your brain that are really the workhorses of how your brain operates and works and controls the rest of your body. And it is such an energetically demanding cell that it needs to make sure its environment is good and that it's not under what we call oxidative stress or inflammation. So, iron has a central role in what happens there. And we have a hypothesis that when the iron response proteins, those that are either allowing iron to be present for the cell to use in its physiological functions, or to store iron when we don't want too much iron hanging around, because too much iron can cause oxidative stress. In order for the cell to regulate that, it has to have certain iron response proteins that either, uh, as I said, make iron present or store it, or actually produce other proteins that can ameliorate any of the issues that are happening with iron. Without going into things like iron Fenton reactions or stuff like that, I guess you just have to take it on faith that the cell does not like to have too much iron around, but it needs iron around in order to be physiologically viable. So it's this constant balance of what's going on. So in dementia, what happens in Alzheimer's disease is these iron proteins get dysregulated, which we think causes the buildup of amyloid plaques and tau tangles. And so this study was looking at, well, if we take a look at the hippocampus, which is a specific area of the brain related to memory, there are subregions of the hippocampus that may respond differently to excess iron and the ability to handle that issue. So that's the motivation of this study. And I worked with undergraduate students and medical students to be able to get the work done.
Okay, great. And I see that a mouse model was used in this study, and I was wondering if you can tell us about the value of using an Alzheimer's mouse model. How applicable is that model, and how useful was the data obtained from it? If you can elaborate on that and the use of such animal models in neurophysiology, neuroanatomy research, I would appreciate it.
Absolutely. We use mouse models to be able to control experimental variables. Um, we can do some experiments in humans, but when we're looking at their tissues, that means we would need to be looking at their brains postmortem, which is an obvious problem. So what we can do with mice is we raise mice from birth to either have specific transgenes or deletions that control for abilities um, or different phenotypes. We can control for different phenotypes in experimental designs. So in this particular experiment, we used a mouse model that has issues with being ApoE4 deficient or ApoE3 or ApoE2, and different um, these different mice handle iron differently. And then
Mhm.
on top of that, we gave them different diets that allowed them or disallowed them to be able to handle different iron scenarios. And then we looked at different ages of these mice. So the nice thing about mice is mice replicate quickly, relatively quickly. And so we can do these timed experiments. They also age faster than humans, so we can have a model for how humans age, and we can do that in a mouse model that's quicker. We can control their genes, we can control their age, and it makes for a nice experimental model. That also means that they're mice and they're not humans, which means that there are some deficiencies in the model that don't quite translate to humans. So that's important to understand as well. So even though we're controlling for certain variables and certain factors in the aging process, sometimes it's not that clean when we go to humans, which I told you from the very beginning are kind of messy.
Yeah.
So we can learn things about physiological processes, but we also have to take it into context that we are working in an experimental model. And while we can look at one variable at a time, humans are very complex. And so we have to translate that a little bit to be able to understand what's happening in humans.
Mhm. Okay, great. And I assume these mice are, by design or by breeding, are genetically what is needed for this particular research.
That's
Okay,
correct. And there's different kinds of genetic models in mice. And the one we use specifically, we're looking at ApoE2, ApoE3, and ApoE4 um, mice.
okay, great. My next question is a specific one. This is regarding one of the figures, one of the graphs that you have in the paper. It's figure 12. I was wondering if you could share that figure with us on the screen here and tell us what it's showing. I looked at it. It's six different panels, and I get the main idea from the legend, but I wanted your input as one of the contributing scientists who have created this figure.
Sure. I thought of something. I'm going to actually share two figures that might be helpful. One of the figures is actually not in the paper,
Uh-huh.
um, but it's good for background. So let me share that first, and then I'll share the graph.
Okay, great.
That way, our audience knows what protein is what. So the first thing is this graph. This is our model for our hypothesis uh, um, in terms of iron regulation. So let me zoom in on what happens normally. So this is a neuron, um, and shows 6 steps. So step one shows iron, which is bound to the iron response protein of transferrin, and transferrin will bind to a transferrin receptor. And this is how iron gets into the cell from the bloodstream.
Yeah.
So iron goes in, and you'll notice it says iron 3 over here, and that talks about the state at which iron is. So we have iron 2 and iron 3. So iron goes into the cell here. It goes into this endosome where iron 3 is removed from the transferrin protein and converted to iron 2. Iron 2 is what the cell needs to use for its mitochondrial functions. But iron 2 can cause oxidative stress if it is around for too long.
Mhm.
So iron 2 goes from the endosome via a divalent metal transporter into the labile iron pool where it's used for mitochondria. But then when it's done, it's stored as iron 3 in ferritin. And ferritin is one of the things that we're looking at. And figure 12 actually talks about another iron response protein, which is called ferroportin. So what happens here is that we have APP, ferritin, and ferroportin iron response proteins to the levels of iron rather than the transport of iron. And here, when they're transcribed, they will produce amyloid precursor protein, ferroportin, and then let's see, APP, ferritin, ferroportin. There we go. We also have microtubules that are held together by tau. When oxidative stress levels goes up, in other words, we have too much iron, this is what happens. And so the iron response proteins are transcribed. We produce more ferritin, and we produce more ferroportin, and we produce more amyloid precursor protein. And what happens here is the iron is actually shuttled out of the cell via ferroportin so that the cell's internal oxidative stress levels can go down. And we use the amyloid plaques in our hypothesis to store iron in the same way that ferritin does until the cell can bring the iron back. And in normal conditions, the amyloid plaques are degraded and iron is, again, brought back into the cell via transferrin proteins. So what happens in Alzheimer's disease is that you notice this X right here. I can't zoom in anymore, but this X right here shows that things like homocysteine can block the ability of iron to be stored in ferritin, which messes up everything else down that cascade. Okay, so the reason why I needed to share that is because Figure 12 talks about the ferroportin story. So that the green proteins that you saw on the cell surface. So let me switch to that diagram and we can talk about the results for that. So what we showed, there's lots of figures, by the way, and we're only focusing on Figure 12 for this particular story. So what I'm showing you here are 4 panels. Remember, these are the green proteins that were on the cell surface that help iron leave the cell, right?
Yes.
So on the x-axis is the time, and that is our time groups. We have 3-month mice, 6-month mice, 9-month mice, and 12-month mice. These are this is a cross-sectional study, so they're different mice at each time point, as opposed to a longitudinal study or a within subject study where we are looking at the same mice but over time. The reason why that doesn't work is because we actually have to sacrifice the mice to look at their brains. So a longitudinal study doesn't work so well in this experimental design. So different mice, and that is a good discussion point about the value of having different mice or whether we should do something longitudinal. On the y-axis is ferroportin, and right here you'll see that this is showing CA1, but then over here this is CA3, and down here is the dentate gyrus, and down here is the hilar region in that part of the gyrus. So hopefully you'll get a chance to take a look at the images that are at the top of the paper that show the different regions of the hippocampus, and if you want, I can go back to that. But for right now, this bottom line of the story is if we talk about that green protein that was on the surface of the cell, the amount of ferroportin goes down, generally speaking, from 3 months to 6 months to 9 months, and then all of a sudden it goes back up. And this is a trend that happens in all of the hippocampal subregions where we measured. So CA1, it goes down, then it goes back up. CA3, it goes down, then it goes back up. The dentate gyrus, very subtly, down and then back up. And then in the hilar region, down and then back up. So that decrease in ferroportin is only significant in the hilar region of the hippocampus. So between 3 months and 6 months, we have a significant decrease in that protein, which means it could mean that we don't need as much. And so we're able to regulate our iron fine with ferritin. And so we don't need as much ferroportin. But wait a minute, what happens from 6 months to 9 months in the hilar region? All of a sudden that ferroportin goes up, which tells us that we are desperately trying to get rid of iron inside of the cell because we're not able to regulate our labile iron pool as well. So that means that somehow ferritin is failing in its job, and we're trying to kick iron out as quickly as possible. And even as we get to 12 months, that trend is still going up. If we looked at mice further than that, maybe the trend would keep going up. We don't know. We didn't measure that far. And so that would be also a nice discussion point to decide whether, well, maybe we should look at, say, 24 months, right? Or something in between to see if that trend continues. So that's what Figure 12 shows, and what would also be a good discussion point for the students would be, do we see the same trend in the other proteins, and how do they work together, looking at that theory of iron regulation that we have. And I'll just put that back up here
Mhm.
again. So from 3 to 6 months, it kind of looks like this, or yeah, 3 to 6 months, it kind of looks like this. But then from 6 to 12 months, we get a lot of green proteins
I
that are
see.
hanging out here. So the cell is desperate at this point to get rid of its iron.
Okay. That's great. Well, thank you for all that. This is very interesting. I think the only exposure I have to transferrin and ferritin in the physiology class is the recycling of iron from dead and tired RBCs. And so this was enlightening that we are connecting these receptors and carriers and recycling of iron elsewhere in the body. At this point, John, if you don't mind that we shift gears a little bit and move away from science per se in the paper and talk about you as a scientist. When and how did you know that you wanted to be a scientist?
Um, yeah, you know what? That is a complex question, and I'm going to give you a slightly complex answer. So, uh, so I think it's helpful for everybody to know that science was the last thing I wanted to do.
Uh-huh.
Um, when I was in junior high school, I saw science as this lab rat type of job where I would sit behind a bench and pipette all day, and that is not what I wanted to do. Thank goodness that's not what I do now. Uh, and so I actually was a pre-law major. I wanted to
Mhm.
go to law school, and I was able to get into a three-year law school program. That means that you do 3 years of undergrad and then 3 years of law school. It's kind of like a 7-year med program where you go 3 years undergrad, then 4 years med school. But my parents are poor. We didn't have the resources to be able to send me to that prestigious school. So I didn't go. I didn't go to law school. And so my parents told me, well, you've got a choice. You can either join the military that can pay for your school, or you can go to a state school. And I grew up in the Pacific Northwest. I grew up in Oregon specifically, and the last place I wanted to go to was University of Oregon. Nothing against the Ducks, but I just didn't want to go there. I was more interested in Seattle, and so my dad said you can go to University of Washington, but then you'll have to establish in-state residency. That's a whole nother story. But when I got to University of Washington, I started as a pre-law major. I was a communications and economics major, which those two topics I still love. I still love the law, and so I'm one of those law geeks kind of thing. But I fell in love with science because I had a biology class where we did muscular stimulation in a physiology experiment on frogs, and we got to see the muscle contract, and I was hooked. I was totally hooked. Why didn't we do any of this in high school? You know, it was, I mean, we had a frog dissection, but we didn't do anything with it. And so, but to do these electrophysiological experiments was so cool.
Mhm.
And so I decided to switch my major. And science was hard for me. It was
Mhm.
really hard. I had to switch from answering exam questions on essays to answering multiple choice questions. And every answer was the same to me. I mean, pick an answer, right? And so multiple choice questions were super hard, but I was determined because I loved it so much. And I dove into neurophysiology and I dove into neuroanatomy, and I really loved the neuroanatomy part more. Ironically, I gravitated more towards that than the physiology because I was interested in circuits and how the brain works as a big system.
Mhm.
So
Mhm.
uh, what do all science majors do in college? They applied to med school. So that's what I did. I applied to med school. I got on the wait list the first year, and then I decided to apply again after I took the MCAT once, and I thought I'm never doing that again. And so I got on the wait list the first year, and I decided not to take the MCAT, which was a mistake. But in retrospect, it launched me on my career, right?
Mhm.
So not all failures are bad things, right? So um, I decided to apply to graduate school while applying to med school again at the same time. And I
Mhm.
got into graduate school without even an interview. So
Mhm.
I I decided, okay, well, I'll do that. And then later, after I got into grad school, I found out I got on the wait list again for med school. I said, well, maybe this is a sign. So I went to grad school and I went to Boston University. and joined the anatomy and neurobiology department there. Same department where I'm at now, which is another part of the story. But I went to grad school there. I started as a master's student because I still couldn't quite let go of the med school thing. But then while I was a master's student, I just realized this was my place. So I appealed to become a PhD student. So I bypassed the master's, ended up getting my PhD. And I worked in a lab with Dr. Doug Rosine on the effects of aging on monkey brain structures. And I used MRI as my measurement to see how structures changed in the monkey brain over time. Um, and so I graduated, and believe it or not, throughout that whole time, I still didn't know if I belonged in science. I
Oh my
still didn't
goodness.
know if it was my thing. It wasn't until my postdoc, where I went to Massachusetts General Hospital at the Athenula Imaging Center, where I worked with other scientists who were interested in questions of MRI in humans this time. We were working on a project in schizophrenia. And I was working with psychiatrists, psychologists, engineers, physicists, and I realized as an anatomist, this is my calling. This is
Mhm.
what I want to do, where I can bring people together to do multidisciplinary science. And it was at that moment I realized this is for me. So I went through all that training, not sure if this is what I wanted to do in my life. And
Mhm.
then when I got there and did my postdoc, I realized this is what I wanted to do. But one of the things that I think is also important, and I'm not sure if you're going to get to this or not, but during that time, I also found education to be something that was important to me. I wanted to help other people learn how to take a multiple choice exam, and because I had so many problems with it myself. And what I realized along the way is that it's not just the ability to take a multiple choice exam that's helpful, but
Mhm.
that faculty aren't necessarily very good at
Mhm.
writing multiple choice exams. So one of my education pursuits is more faculty development and helping faculty write good questions and helping students answer those questions. And then I really expanded into service learning and outreach and studying how effective learning outside of the classroom is for students. And then now in my role at the medical school, I also focus on curriculum and how we can make curriculum better so that students can have a good educational experience. And I'm doing these two things at the same time. I still have a neuropathology lab, and I mentor students in my lab, and then I do education at the same time. So all of this really started at the moment when I was a postdoc, when I realized, this is what I want to do.
Well, that's great. And in many ways, you answered parts of my next question, which
Oh,
was
sorry, I didn't mean to go ahead.
no. That's quite all right. I'm glad that you went through that narrative because I was going to ask you about the path that has led you to your current job. So in many ways, you have answered that, unless you want to elaborate a little bit more on that. Um,
Um, I can
did you
speak I can speak specifically to my current job. Right now, I'm the co-director for the basic science course in our preclinical medical curriculum for the med students and the PA students. So I work with in a leadership role with a lot of different module directors and classroom instructors and lab instructors, and I organized that that whole machine. Um, so I work with a hematologist who's the clinical side of that course, and it is very akin to me serving as an anatomist in science, as
Mhm.
an anatomist in education. I'm also a hub for organizing the educational experience for the students. So that's the large part of my work. And then, like I said, I'm still mentoring students in my lab, working on Alzheimer's disease things and teaching and learning.
Yes, and I know about several other functions that you do through the Human Anatomy and Physiology Society and possibly other professional societies.
Yeah.
And that's, I guess, the life of uh, a professor in academia in many ways.
Hmm. It's a lot of fun. It really is, as you know.
Well, thank you, John. This was really interesting. I think I have another question for you that's also away from science, but nevertheless, I'm interested in hearing your take on what is it like being a scientist who identifies as Asian and being the first in your family to earn a PhD?
Hmm. Yeah. So I think many of the students that you're teaching might know that there are a lot of Asians in science, but there aren't a lot of Filipinos.
Hmm.
And so I'm Filipino specifically, which I'm very proud of. And I think being a person of color, specifically Filipino in science, I feel a weight of responsibility to be an example to others, excuse me, especially Filipinos who want to go into science, science, medicine, or any sort of health career. Um, yeah, so I feel like there's a responsibility to be a good example. Um, but I think there's also opportunities for me to help diversify the community of scientists, and that's been important to me as well. I don't think there's any specific thing about being Asian per se, other than breaking stereotypes that Asians are quote unquote smart, right? We're all trying to be educated and get better at what we are doing, and I don't think we're any smarter or any less smart than anyone else. So, especially for Filipinos who have that stigma, thinking that they can't make it in the health sciences, we can. I think we just need the right supports. Many Filipinos are underprivileged for sure. And so I think encouragement and helping them with whatever they need to navigate is something that I have done in my career. But I don't limit that to just Filipinos. And anybody who wants to reach their dreams, I like to help students figure out how to do that.
Yeah. Yeah, that's great. I always think of it as when I am chasing a paper to try and find it from, a published source. And the last thing I would think about is who's in the author line and from what background did it, because we are all interested in science and
Right.
then finding out the how does our body work or how do we fix something that goes wrong in the clinical aspects.
Yeah.
Well,
And being the first PhD student to graduate from my family on either side made my parents very proud. Made me proud. And so I'm glad that I had that opportunity.
yes, indeed. Um, and my last question in this interview is regarding, we talked about imposter syndrome, where we sometimes find it very hard that we belong in this field or that. So here I want to ask you about a moment where you felt you really belonged in the field of science.
Yeah, it really goes back to that moment in my postdoc that I was referring to earlier, where I realized that I had a specific role that I could add a lot of value to.
Hmm.
And I think when you think about imposter syndrome, one of the things that helps minimize that, and I'll explain the word minimize in a second. One of the things that helps minimize that is realizing your value. And sometimes that could be an external validation, sometimes that could be an internal validation. But either way, realizing where you're most valuable is something that helps a lot.
Hmm.
That said, one of my pieces of advice that I get to my students that I mentor is that imposter syndrome never goes away. In
Hmm.
fact, actually, it gets worse. As you progress in your career, you are going to become amazing in your own right, and other people are going to become amazing in their own right, And so, comparing yourself with others is a futile exercise because everybody's being amazing in whatever they're doing, and there's no reason to think that you have to be like anyone else or that you're better or worse than anyone else. And so because everyone's becoming amazing, imposter syndrome gets worse as you get older, as you get more seasoned, as you get more experienced. And so I advise my students to become comfortable with who they are and the value that they add in the spaces of influence that they have and be good with it. Now, being good with it is a lifelong skill. There's times when I'm sitting there with my colleagues going, why am I even here? So it constantly comes back. It never goes away. I don't think anybody has ever solved imposter syndrome. And so you just have to be okay with that. That's going to happen. And once you find peace with that, so to speak, even though it never goes away, it is easier to navigate through those difficult situations when you don't think you're as good or as worthy as the next person. And just keep in mind that you are there for a reason and you are qualified no matter what you're doing.
Yes. Thank you for that. I really appreciate it. And with this, I am going to conclude this interview, and I want to thank you again for taking time. I know that you are busy, and especially that we are heading towards annual conference for the Human Anatomy and Physiology Society. So thank you again, Jon. I appreciate this.
Well, thank you, Burhan. I appreciate it too.
All right, great.